Transgenic production in plants, part 2. Previous video I had covered the basic concepts of gene and traits, identification and isolation of gene for specific traits. Now the next is designing of gene constructs for transformation. Here is a schematic representation of the transgenic construct. So you have a selection marker gene, promoter, transgene, as well as the termination sequence. In brief, let us cover promoter, selectable marker genes, termination sequence, and gene of interest, or the transgene. Once a gene has been isolated and cloned, cloning is done in a bacterial, that means it is amplified in a bacterial vector. It must undergo several modifications or manipulations before it can be effectively inserted into a plant. So a promoter sequence must be added for the gene to be correctly expressed. Now a promoter in a transgenic plant is a DNA sequence that regulates the expression of a particular gene. It is an on and off switch that controls when and where in the plant the gene will be expressed. Okay, there are three categories of promoter: constitutive promoter, tissue-specific promoter, and inducible promoter. So many times currently the most promoters in the transgenic crop varieties have been the constitutive promoter. So that means it is causing gene expression throughout the life cycle of the plant in most of the tissues. So this is the one that is a cauliflower mosaic virus isolated promoter, that is CANV35S, is the promoter that is usually used, and it results in a very high degree of expression in plants. And some other promoters that are used is tissue-specific promoter, that is a seed-specific promoter, like the Visilin promoter from P. Visilin storage protein gene. Next is an inducible promoter. That means to say that it responds to the plant's internal or external environment, such as light inducible promoter. The promoter from the CAP gene encoding the major chlorophyll A and B binding proteins. So a selectable marker gene is added to the gene construct. Basically, these are used for the confirmation of gene expression in the transgenic plant. And this is essential as a part of the gene construct because achieving incorporation and expression of the transgene in the plant cells is a very rare event, and only few % of the targeted tissues or cells it occurs. So these marker genes are essentials. Marker genes can be scorable marker genes or selectable marker gene. See, the scorable marker gene expression is easily detected either through highly sensitive enzyme assays. One such example is the NOS, napalin synthase. Next is the selectable marker gene. The selectable marker genes, they encode the proteins that provide resistance to agents that are normally toxic to the plants, like antibiotics or herbicides. So only the plant cells that have integrated the selectable marker gene will survive when grown on a media containing the appropriate antibiotic or herbicide. Now here the example is NPT2, that is neomycin phosphotransferase. Okay, so this marker gene, like any other gene inserted in the construct, also requires promoter and termination sequence for proper function. termination sequence. This sequence signals to the cellular machinery about the end of the gene sequence. It influences the rate of transgene expression in transgenic. Gene of interest or transgene. The gene of interest has to be modified sometimes to achieve expression in plants. Let us see an example. In Bt gene, a minor modification of AT nucleotide to GC nucleotide pairs for enhanced production of gene products in plants. Now, here, the Bt gene is for insect resistance, is of bacterial origin, and it has a higher percentage of AT nucleotide pairs compared to the plants which actually prefer GC nucleotide pairs. So, a minor modification by substitution of AT nucleotide with the GC nucleotide in the Bt gene is possible without significantly changing the amino acid sequence. And of course, and this modification results in an enhanced production of gene product in plant cells.